Dejah, thank you so much for joining us on MD Newsline podcast. Can you please introduce yourself to our audience?
Of course. My name is Dejah Blake. I'm a fifth year PhD candidate at Emory University. And my PhD work is in engineering CAR T cells and looking at it from the standpoint of engineering, but also looking at it from the standpoint of how can we make CAR T cells work better for our patient populations.
Fascinating. Tell me a little bit about what your impression of the ASH conference has been so far.
Ash is super fun, super tiring because there's just so much to do. I think it's like a kid in a candy store or like analysis paralysis where you're trying to figure out where to go first, what to do first, all that. But it's been great. You learn a lot, you meet a lot of people. People have been really nice. I think it's a really good place to come as a trainee, especially like taking the next step in my career and kind of figuring out what I want to do or like who to talk to. It's been
Hmm.
great.
Wonderful. And tell me a little bit, have any sessions in particular caught your eye? Any speakers or data results that caught your eye?
No, definitely. So I've been, I feel like obsessed with in vivo CAR T cell manufacturing. So definitely, I think the session was on Saturday. Saturday morning, there was a session on in vivo. And there was some in vivo CAR T cell talks about it and using gene editing to transduce T cells.
Mm.
So I think it's the next forefront when we think about CAR T cells. So it's just really cool to I think there was one about from Orna Therapeutics that was pretty interesting to me.
Why is that particularly fascinating to you? What about it kind of piques your interest?
The biggest part about it that piques my interest is the accessibility. So CAR T cell is what we call the boutique drug, where patients' blood are apheresed, engineered, or T cells isolated, engineered, then given back to the patient. So it's a very personalized drug in a sense. The special sauce of it is the manufacturing part. Um, in vivo CAR T cells gives an option for an off the shelf product. Of course, they have other off the shelf products with say CAR NK cells or allogeneic CAR T cells. But I like the part of an off the shelf viral vector that can be given to a patient pretty quickly, um, and takes away the waiting for the T cells to be engineered and all that stuff. But also, it's a very expensive process. So I think the biggest part is the accessibility. Also, the science is really cool. I think gene editing is very cool. It's even beyond cancer. It's given a chance of life for many people who might not have it. But it's the accessibility that really.
Yeah, I was gonna ask about price specifically, so I'm glad you mentioned that.
Yeah. So of course these viral vectors are expensive. They're definitely expensive. However, when you think about what the regular boutique CAR T cells, those are inherently going to be more taxing on the companies, therefore taxing on the patient. So I think in vivo CAR T cells, I like or using viral vectors sort of gives a chance for patient accessibility and maybe a bit cheaper in the grand scheme of things.